Welcome back, everybody. This is part 2 of the lecture on population genetics. In the first part, we mainly focused on frequencies of genotype, of phenotype, and allele frequencies. And again, that just kind of sets the stage for us to understand how variable individuals within a population are, and that helps us understand how readily they're able to adapt to a particular environment. And so that's mostly what this lecture is about, is we're going to look at those mechanisms, those processes that can occur that allow us to get new alleles and adapt to an environment. Okay. The previous lecture, we also looked really briefly, and you're going to do a example in the lab portion of the class of genetic equilibrium, what it's like when a population is not evolving, when allele frequencies are not changing, when it's stable, right? But we know that natural populations actually don't experience genetic equilibrium. There's actually always changes that are occurring, and those changes are occurring at the gene level, and we refer to that as microevolution. And so microevolution are these changes in alleles or genotype frequencies within a population over generations. Okay, and we're going to look at the processes that do that. So this picture is really kind of depicting, you can see the first generation. These would be like your parents, and you can see in that first one, this beetle has this green color, and in the second one, the beetle is more of a multicolor with the green on one side of his body. And you can see when they mate, there's going to be a different combination of those two alleles that are being split into the different eggs that the female can make and the different sperm that the male can make. So each one of the egg and the sperm, they're only getting one copy of the two alleles that they possess. And so if you're heterozygous, then you can you possess two different copies, and you can see then that would end up as a different combination in the offspring there in the second generation, and you see how those phenotypes get expressed. So again, this is why some of your siblings may look really a lot like your parents, and others may not look so much like the parents. It's just that different combination of the alleles that they're receiving. And you can see that expressed in some of those other pictures there. So this type of microevolution really results from 5 microevolutionary processes, and that's what we're going to go over. The 5 microevolutionary processes are non-random mating, mutation, genetic drift, gene flow, and then also natural selection, and we're going to see that there's different types of natural selection. For mutation, all I want to say on this subject is that mutation is the only way that we can actually get brand new alleles. Okay, so alleles are the DNA that we possess, right? And so in order to get a new allele in a population, then something has to happen to the DNA, right? You can only have an allele in the population if an individual possesses it, right? So how do you get something brand new, something novel? Well, that happens in the DNA, in the creating of the DNA in our cells. And not really the creating, but all our cells are dividing. And so when our cells divide to make an egg or a sperm, as the case may be, we need to put a copy of our DNA in that egg and sperm. And so our DNA actually duplicates itself and then divides. And it's in that process of duplication, duplicating the nucleotides that make DNA, that we actually make mistakes sometimes. And those mistakes can either be harmful, it can actually render the DNA unusable, and so we would then see that allele die out, right? It could make an allele that's advantageous, though. Maybe it codes for a protein that causes you to be more immune to a specific DNA. So that would have a positive effect. Most often mutations are neutral. because there's so much redundancy in our DNA that when a mutation occurs and a nucleotide changes, it doesn't really change the protein that it's coding for. So that's a so again, mutation is usually neutral. Mutations happen all the time as we're making new cells in our body and as we're making egg and sperm, but usually it's neutral. But sometimes you get that positive aspect that can actually allow you to adapt better to a changing environment as well. Okay, so we're going to be talking more about mutations as we get into more specifics on the different types of species that we have in the world. So I'm going to go on and talk about one of the other microevolutionary processes referred to as genetic drift. Okay, so genetic drift, excuse me, refers to gene frequencies that are changing over generation to generation, but the change is just random. Okay, it's a matter of chance that these gene frequencies are changing over time. Okay, this means that an allele can actually be eliminated from a population just purely by chance. Okay, the resulting changes in that allele frequency then occur regardless of the fitness of the individuals. Okay, so again, if it's by random chance, it doesn't mean that the allele wasn't helping the individual. So it really just doesn't matter the fitness of that individual. Fitness of an individual in this sense refers to how well adapted an individual is to their environment. Okay, so the individuals could be very well adapted, but it's just, again, random chance that's causing that allele to disappear. Okay, so if you can't wrap your head around that, realize that some individuals have bad luck and they just can't find a mate. If you can't find a mate, then you can't pass your genes on. Okay, and that happens. And we'll look at some other examples too. But over the long run, what happens is that genetic drift actually favors either the loss of an allele or the fixation of allele. But the number of generations for either the loss or the fixation to occur really depends on the size of the population. So this phenomenon will happen faster in smaller populations. So let's go over the graph so that you can understand this a little bit better. So this graph, always when you're trying to understand a graph, you've really got to look at how the graph is labeled in order to understand what it's trying to show you. So up on the y-axis, that's frequency of a particular allele that they're referring to as capital B. So it's the dominant form of this allele, which looks like fur color. So it might be that it's black, okay, or brown. So that's the frequency that the dominant allele of fur color occurs in the generation. And over the x-axis, it's showing you the different generations over time. So we have 10 generations later, 20 generations later, 30 generations later. Okay. So the graph here is showing 3 simulations of genetic drift in 3 different population sizes over 50 generations. So at the beginning, all 3 populations have identical allele frequencies. Okay, half have the dominant allele and half have the recessive allele. So they start at 0.5. So in the small populations, the gene frequencies are fluctuating greatly in each generation. How do you know which line is depicting the small population? Well, you see the little notation next to them. Capital N equals, and there's a number. So remember from our population dynamics lecture, capital N is the symbol we use to depict population size. So in each of the red lines above and below, the population sizes are 10. In the blue line in the middle, the population size is 1,000. So again, in each of the small populations, you see the red line going up and down pretty quickly. That means the gene frequencies are fluctuating greatly in each of the generations. And eventually, it's either eliminating all the black fur individuals, or at the bottom at the bottom it's eliminating all the black fur. At the top, it's eliminating all the white fur. Okay, and that's just by random chance. So now your population is becoming what we call monomorphic with respect to fur color. It means they're only displaying one color of fur. Okay, so when you have less individuals to mate with, and you start with 50% of the alleles being dominant and 50% being recessive, when you have less individuals to mate with, you have less of a random chance to mix up those alleles. Okay, so what's happening in the upper population is that it may just be by chance that more of the individuals with the black fur are mating with one another, and they're passing on that trait. Okay, again, it can just be chance, bad luck. The ones with the white just happen to be in the wrong place at the wrong time. Okay, same with the lower one. Okay, in the large population, in the middle though, the frequency of alleles would fluctuate much less because there's much more individuals and much more of a chance to encounter one another. Okay, so if you have a greater chance of encountering one another, you have a greater chance of mixing up those genes, of encountering different individuals with a different allele type than what you have, or genotype than what you have. So genetic drift can cause allele loss or fixation eventually in a large population, but it just takes much longer. Okay, so it's going to take a lot longer than 50 generations to do that. Okay, so we see this when a population size dramatically decreases, then genetic drift can quickly alter allele frequencies. Okay, and a population size can dramatically decrease in two ways, and refer to those two ways. One is bottleneck, and the other is founder effect. So this picture indicates what a bottleneck is. So a bottleneck is that suddenly the population gets dramatically reduced by something out of your control. It could be an earthquake, a flood, a drought, disease. So it's showing, what this picture is depicting, it looks like two bottles kind of attached to one another. At the top is the original gene pool. Okay, and you can see the variability in the alleles based on the different colors. Okay, and so in the skinny portion is where something happened. Something happened, maybe there was a drought, there was a disease that eliminated the food supply, or it killed several individuals. So just by random chance, you can see that square in the middle, it depicts that there's a very small number of individuals, and the individuals who died out just happened to be the ones with those other colors of alleles, the pinks and the orange and the yellows. What's left, the ones who survived, just happen to have these other alleles, the blues and the greens. So they're the only ones left that are able to reproduce. And so they do reproduce, so their surviving members were able to reproduce, but they can only pass on the alleles they have. So now the new population has less genetic variation. Okay, so again, we lost alleles, that's genetic drift. It happened from random chance, but it happened from a population bottleneck. The population numbers were dramatically reduced due to some kind of environmental condition, a condition that was out of control of the population. There's a picture of a cheetah there because this is what's happened to them. We had a robust cheetah population in Africa, and then due to several things, disease, hunting, things like that, habitat loss, the population number is greatly reduced to very few individuals. But through protection now, they've been repopulating and their population size are large. But when we look at their genetic variation, it's really low. The genotype frequencies or allele frequencies are very, very low. They don't have much genetic variation. And that's a problem because if there's a disease that sweeps through this cheetah population and they don't happen to have the allele to fight it, then we could just see the extinction of cheetahs. And so cheetahs that are part of the zoo programs, we actually have mapped out each of their genotypes. And so they're put into a database, and it's like a dating game. And so when a zoo is able to breed one of their cheetahs, they won't just randomly pick a cheetah to breed with. They actually go to the database and they look for a cheetah that has genes that are much different than the cheetah that they have. And so therefore the offspring will display a little bit more genetic variability. And so that's one way that humans can kind of intervene and try to help increase genetic variability. So let me give you another example of a bottleneck, just in a different way. So here in the top picture, it shows you a pond with frogs in it. And you can see there are 3 phenotypes of these frogs. There's green, there's yellow, there's striped. These are all the same species because it's a population of this frog. Well, they experience a drought. And when they experience a drought, the pond water dries up, and that's less space and less food for the frogs to survive. And just by chance, so that's the bottleneck, it decreased the population size, and by chance, the green phenotype just didn't survive. And so it's not necessarily, in this case, it's not because of an adaptation that the brown or the yellows had to the drought. It's just that maybe the green frogs were hanging out at the edge of the pond when it really started to dry up and they couldn't make it back to the water. So just by chance, the green individuals that had the alleles for green died out. And so, but then the rains came back, and you can see in the lower picture, the rains came back, the pond filled up, and the individuals who are left to mate don't have the green allele. So now all the individuals come out yellow and brown. Okay, so the middle picture is what's really depicting that bottleneck. Again, bottleneck has a skinny portion. So think of a bottle, it's wide at the bottom and then it comes down into a skinny portion, real thin portion. And so that's why it's named that. We're using that as a picturesque way to describe a large population suddenly becoming a small population. So again, individuals die out. It's the individuals that possess the alleles, and so their alleles die with them. Okay. Here's a local example of a bottleneck that was experienced. These are northern elephant seals, local as far as like Northern California and up. So if you go and go to the northern side of California, you might encounter elephant seals there. They, due to hunting for their blubber as a source of oil, they were hunted from tens of thousands of individuals down to 20. Their population went down as low as 20 individuals. So you can imagine if you had tens of thousands of individuals and they were killed off to just 20 left, all the genetic diversity that was lost. So the population that exists today, those 20 individuals were able to mate due to the protections that we offered them. And now their population size has recovered to over 100,000. But again, their genetic variability has been so reduced that there's a worry that if something happens in the environment, that could easily wipe out the population because you don't have the genetic variability or the random chance to be able to survive whatever that change in the population is. Now, if they can survive over generations and generations, yeah, their genes are going to evolve over time and we might see an increase in genetic variability. But we really have to protect them. in order to allow that to happen. It takes a long time for that to occur. Okay, another causes of genetic drift is founder effect. So founder effect also is the population size decreases, but this is due to a proportion of the population getting up and migrating and starting a new colony of individuals. Okay, so this doesn't have to do with random chance from a environmental event. This has to do with the random individuals that get up to move to find a new place to live. So it's called founder effect because the small group of individuals are finding, or they found, a new area to live. Okay, so again, you can see from the pictures of using butterflies that just by chance only the individuals with the orange and the black alleles migrated out, and the white ones stayed behind with the rest of the population. So now you can imagine with the new population, those individuals, as they mate with one another, the white allele doesn't exist in that area. So you're going to see a change in the allele frequencies there. And it's possible that that new allele frequency helps them adapt to that environment a little bit differently, right? And over a much longer period of time, we can actually see a new species occurring if the genetic frequencies change more and more. So in this case, with this new population of founder effect, the allele frequencies in this founding population, they might differ markedly from that original population. Okay, so it's similar to bottleneck, but the reasons for the smaller size in the population is different. Okay. Okay, another concept we want to consider now is gene flow. Because when we talk about populations, we said that populations are, the definition is individuals of the same species in a particular area. So you can have the same species but a different population somewhere else. And we call it a different population because the two populations are generally isolated from one another. And so if they're isolated, they don't mate with one another. And so therefore they're only mating with each other within their own populations, and then they're only mixing the alleles that they have within that population. But with gene flow, this can occur by chance, is that maybe a few individuals manage to migrate to another population. Okay, maybe the normal situation is they wouldn't cross whatever barrier is there. But once in a while, some individuals do, for whatever reason, we don't know. Okay, and if they do, they might bring the different alleles with them. So this picture is showing a really extreme example there. This wouldn't happen in nature, but you've got two populations of birds separated by this waterway. I don't know why the birds have a hard time getting over the water, but maybe there's a predator down there that eats them. So again, just a simple example. But the point is that in one population, it's showing they have all the recessive alleles. They're homozygous recessive. In the other population, they have all the homozygous dominant alleles. Okay, they're still the same species, but they just are possessing a different form of those genes, right, in the expression of the alleles that they have. But look, one guy made it over. He was able to fly over. Maybe it's a long distance over that waterway, and most individuals need to stop and rest, but they can't rest on the water. But this guy didn't. This guy made it. And so if this individual can mate with the others, now you can see that you would get heterozygous individuals in that population. So gene flow just means the exchange of genes between two populations. It's usually a few individuals that are able to do it, cross whatever barrier that's separating the two populations. But when that gene flow happens, it's usually a good thing because it increases the variation of the genes in that population. So again, the more variability, the more likely it is the individuals can adapt to their area if the environment changes a little bit. Right? If enough gene flow occurs, if maybe those birds are going, hey, that guy did it, we can do it, and there's more gene flow, even from both directions, then the two gene pools may end up becoming genetically similar over time. Okay. Human distribution is an excellent example. So there was a time where, we humans, we did not have all the technology that we have today. So we had people who were distributing around the globe by walking or building canoes and things and risking their lives to cross the ocean. And so when human populations started to move around the globe, and we have really good evidence that human species started in Africa, and it was due to drought and things that a subset of the population, founder effect, got up and migrated and started crossing into Europe, crossing over, and they split over into Asia. And as they would stop and find a new area to live, find a place with good resources, their population, they would mate, and their populations would increase. And then as populations started to get up and move again to explore, then two different populations would encounter one another. But they might have looked differently because each individual population had their own set of alleles. So they might have looked differently to one another as far as skin color, hair color, facial features. And so they might have seen each other as different, right? But now, as we're in more modern times and it's easier for us to visit different populations, we're not so isolated in this world anymore, and we're starting to see that our allele frequencies are starting to homogenize within the world. And we know that we are all one species. We're not different from one another. There isn't a lot of major genetic variability enough to determine different human populations are different species. The genetic variability only accounts for just differences in phenotype rather than differences in species, or what we might call race. Okay, so humans are a really excellent example of the gene flow that occurs amongst groups of people.